Hi folks, and welcome to another edition of Zog Science. Today we are going to be building on our discussion about membranes by talking about the process of diffusion. So passive transport is diffusion of a substance across a membrane with no energy investment. Diffusion is specifically the tendency for molecules to spread out evenly into the available space. So, although each individual molecule is moving randomly, diffusion of a population of molecules may be directional. So, in general, you can have the diffusion of a population of molecules going in a specific direction, even though each individual one is moving backward or is not necessarily moving in that direction. The whole point of diffusion is to reach dynamic equilibrium. And dynamic equilibrium is where you have as many molecules crossing the membrane in one direction as they are in the other. So this is what diffusion is, right? It's the random movement of molecules. So they're just moving about randomly in order to achieve equilibrium. And if you'll notice, right, we have a higher concentration at the top and a lower on the bottom. So when there is a membrane, you have molecules that are going to be moving across the membrane and some that are moving back. But in general, more of them are going to be moving across the membrane in order to reach equilibrium. Same thing can happen with specific molecules like oxygen and carbon dioxide. So diffusion can be occurring between just one solute, right? Or it can be occurring among many solutes. So in our first example here, we have a higher concentration on the left than on the right. Overall, we're going to have a net movement of the molecules to the right until we reach equilibrium. Same thing for the second scenario where now we've got two different molecules. One's on the left, one is on the right. Over time, they're going to begin moving back and forth until we reach a equilibrium for both molecules. Now, substances diffuse down their concentration gradients, where you're moving from an area that is more concentrated to an area where it's less concentrated. This requires no work. This is actually moving with this idea agrees with the second law of thermodynamics, right? Because it is a higher level of entropy when your substances are more spread out, when the molecules are more spread out from each other. And therefore, we don't have to put energy in, we don't have to put work in order to make this happen. The diffusion of a substance across a biological membrane is called passive transport because no energy is expended in order to make it happen. Now, osmosis is a special type of diffusion, which is diffusion of free water across a selectively permeable membrane. Water diffuses across the membrane from the region of lower solute concentration to the region of higher solute concentration, and that is because the water is in a higher concentration where there is less solute, and there is less water where there is more solute. So the osmosis is going to be occurring in that particular direction. And recall that osmosis of water takes place through the aquaporins because water itself cannot move across the cell membrane. So in this example, we have a higher concentration of solute on the right, a lower concentration of solute on the left, which means that there's more water on the left and less on the right. Therefore, the water is going to move to the right in order for there to reach equilibrium where they have equal concentrations. Water has specific words. So tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. And there are three different ways we can describe solutions. We can talk about an isotonic solution, which is where the solute concentration is the same as it is inside the cell, which means that we're already at equilibrium, so there's going to be no net water movement. We can also have a hypertonic solution. This is where the solute concentration is greater than that of inside the cell, and so therefore the cell will lose water because the solute concentration is greater outside. Or we can have a hypotonic solution. That is where the solute concentration is less than it is outside the water, therefore the cell will gain water. We can see some examples of what would happen in an animal cell. So in an animal cell, when you place it in a hypotonic solution, water comes in. Because it is just a cell membrane, if you have too much water that comes in, it will cause the cell membrane to break. If we place it in an isotonic solution, then it will be normal, right? Water is going to be coming in and out. And if you place it in a hypertonic solution, it is going to lose water. And again, because there's no internal structure there for the cell itself is going to shrivel up. Now, cell walls help maintain water balance. So for plant cells, they, if placed in a hypotonic solution, will swell, but they will not burst because they have the cell wall that helps them to maintain their structure. If they are placed into surroundings that are isotonic, there's no net movement of water in or out of the cell will become limp and the plant may wilt. And that's because there's not pressure from the water pushing out on the cell walls. In a hypertonic environment, plant cells will lose water, and that means that the cell membrane will actually pull away from the cell wall, which leads to plasmolysis, which is a lethal effect and will generally cause the cell to eventually die. So here we can see what it looks like when it's turgid, right? It's got nice pressure on it, it's pushing out on that cell wall, helping the plant to maintain its shape. When it is isotonic, it is going to be flaccid. It's not going to be, the plant's going to be wilting a little bit. And then if we don't have enough water at all, it will be plasmolyzed. You can see plasmolyzed, you see how the cell membrane is pulling away, and that is going to probably lead to the cell and eventually the plant's death. Now, facilitated diffusion is a special type of diffusion, and this is passive transport that is aided by proteins. So in this case, transport proteins, which as we learned about before, can either be channel or carrier proteins, help speed the passive movement of molecules across the plasma membrane. Typically, these are going to be channel proteins, although they can be carrier proteins as well. And they provide basically corridors that allow a specific molecule or ion to cross the membrane. Aquaporins, remember, are the specific ones that help with the diffusion of water. There can also be ion channels that open or close in response to a stimulus, which can sometimes be called gated channels. We will talk more specifically about gated channels when we get to our discussion of the nervous system later in the year. So here's the function of aquaporins. You can see how it is providing an avenue for the water molecules to move in and out. Because this is, of course, a random process, these water molecules are just kind of randomly bumping back and forth between them. And you can see how the aquaporin is allowing them to move in and out through the middle of its structure, which is a hydrophilic structure on the inside. It's hydrophobic out here on the outsides so that way it can interact with the cell membrane appropriately. Carrier proteins undergo a subtle change in shape that translocates the solute binding site across the membrane. So when this change occurs, it moves the molecule into the cell or out of the cell, and no specific energy input is required. So here is a channel protein or a diagram of a channel protein. You've got your solute that is just moving through it. You can also have a carrier protein where the solute will come and it will bind. This causes a change in the shape of the protein, which is then going to allow the solute to move into the cell, or it could be out of the cell as well, depending on the concentration gradient. That's all I've got for you this time. Thanks for watching, and I'll see you next time.